All right, let's talk a little bit about biochemistry or the life's chemistry, things that you see in biology that are related to our survival as an organism, whether it be plant, animal, fungi, bacteria, and so forth. This is the chemistry of life. So it's made up of carbon, hydrogen, oxygen. Those are the main components of the chemistry of life or biochemistry, with additional things like nitrogen and phosphorus and sulfur. And of course, iron occasionally too. Let's talk about the major macromolecules. These are what make up organisms, and those are carbohydrates, lipids, proteins, and nucleic acids like DNA and RNA. So these are things that you should be at least a little bit familiar with, right? I mean, these are the things that we eat when we have food, or what we're made up of. So we're talking about organic chemistry, chemistry being made up of carbon atoms and so forth. That's what we mean by organic. Now, when you hear the word polymer, you might not always think about what does that mean actually? Well, polymers are basically large, important molecules, often biological molecules, depending on what type of polymers we're talking about. And they are constructed by basically joining smaller molecules together, and those are repeating and are considered or called monomers. So polymers are basically just bunches of monomers joined together, the same kind of piece over and over again. Now, this is done through biochemical enzymes and things like that, where you'll join different monomers together. And if we do so, we call that dehydration synthesis. As you can imagine with the word dehydration, that means it has something to do with water and being dehydrated. So it's actually pulling water, maybe from two sugar molecules or two amino acids that make up proteins, joining them together, those two monomers, to make a polymer. Now, hydrolysis is the opposite in the sense that we're breaking down two monomers. So we're making we're breaking pieces off of a polymer. And again, we're using water to do so. So hydrolysis means that water is being added to the enzyme. Again, enzymes are proteins that speed up chemical reactions. So chemical reactions could happen on their own, but enzymes make it work incredibly faster. Now let's talk about carbohydrates. As the word itself should suggest to you, carbon, carbo, hydrate, water. So we know that it's going to have carbons and H2O. When we talk about atoms, we write large letters representing them. So C would represent the carbon atom, H would represent hydrogen, and O would represent oxygen. And if you ever seen the chemical formula for water, it's H2O, where we subscript H with a small little 2. So that would mean that there's two hydrogen atoms. So when we think about carbohydrates, you should think of it as being C, H subscript with a little 2. And oh, it's not shown here, but that's what essentially it is. It's water plus carbon. And as you can imagine, carbohydrates are important for energy. Simple sugars, for instance, will make you move around, have that extra energy source. You know that from your own personal experiences. But carbohydrates are also important for structure. Plants, for instance, are made up of cellulose, a polymer of carbohydrate monomers that make for rigid stems and leaves and so forth. So again, the monomers for a polymer would be a monosaccharide representing a simple sugar. So monosaccharides are simple sugars containing 3 to 7 carbon atoms. And again, it would have this ratio of 1 carbon atom to 2 hydrogens to 1 oxygen. That hydrate is the water, H2O. So carbohydrate. And so here are some different types of carbohydrates. This is the, you'll see like the chemical formula, and you'll actually see the structural formula. The chemical formula, like for glyceraldehyde, is C3H6O3. And then the chemical or the actual structural formula you'll see above it, where you'll have your carbon atoms. By the way, carbon atoms can hold 4 covalent bonds. If you might want to refer to some chemistry lectures or past history of chemistry to remember that. Hydrogen atoms would hold 1 covalent bond, and then oxygen would hold 2 covalent bonds. And so when you see that dash, those lines there, right here, for instance, that is representing the covalent bond at the shared electrons, 2 shared electrons. Now it's not shown between this O and this H, but there is a line there also. It's just kept as a shorthand. And so again, each carbon atom has 4 covalent bonds. That means essentially equal sharing of electrons. Now, the thing about structural formula versus chemical formulas, look at this right here. We have glucose and fructose. Again, these are all monosaccharides. And here we have C6H12O6. That means there's 6 carbon atoms, 12 hydrogen atoms, 6 oxygen atoms. And look over here, fructose also is C6H12O6, but the chemical structure is different than the actual chemical formula. So again, the structural shape is different, and we call these isomers. And so each one of these corners is a carbon atom. They did not draw it because that is shorthand. It's under the assumption that you, as a biochemist, will know that already and naturally that these corners have carbon atoms. This saves on having the right amount. And they do other shorthand where they might not even put the hydrogens and the OHs in, with the assumption that you already know that. But just realize that's why the chemical formula C6H12O6 doesn't tell you everything. You need to see the structural formula as well, or structural shape. Again, this would be shorthand here. You know that a carbon atom would have 4 covalent bonds. So this hydrogen would come out this direction, this hydrogen would come out that direction, and then this carbon would attach to this oxygen, which would then attach to the H. Whenever you see the OH, we call that hydroxyl, by the way. So OHs are hydroxyl groups. And so these are all examples of monosaccharides, simple sugars, mono being 1, and of course saccharide is representing that sugar. And then through dehydration synthesis, and these arrows mean that the chemical formula can go back and forth, it can go in either direction. We can have dehydration synthesis where we remove the water. So we can remove a hydrogen and a hydroxyl group and join this carbon with this oxygen and this carbon with this oxygen. So we've removed 1 oxygen and 2 hydrogens, and you'll join these 2 monosaccharides together, one being, of course, glucose and the other one being fructose. And then together, we now call it sucrose. And that is a disaccharide, meaning 2 simple sugars. Now that's dehydration synthesis. Here's the byproduct, water that's left over. Now let's say you were to drink water and you could break this bond through a chemical reaction, again, using enzymes, because there's going to be a protein enzyme that's helping with this reaction. And then you'll have what you call hydrolysis. Hydrolysis is basically utilizing water to break these disaccharides into 2 monosaccharides. And so that is really good for breaking down simple sugars. Now we have here a polysaccharide. Now this is again representing multiple monosaccharides joined together. And so you have, instead of a disaccharide meaning 2, poly, of course, means multiple. And then this would be examples of polysaccharides. And again, these are complex carbohydrates. And they're made up of hundreds of monomers, and they're all linked together by that dehydration synthesis, that removal of water. And so here you have cellulose, starch, and glycogen. Cellulose is what we find in plants. Starch we also find in plants. And then glycogen we actually find in the liver of animals and the muscles of animals. And so this is a polysaccharide that we find in animals, and these two would find in plants. Cellulose gives the plant structure, helps it to stand tall, while starches provide energy. We as humans can't digest cellulose. Cows can, but that's because they have a symbiotic relationship with bacteria. But we can digest starches like we find in potatoes and flour and bread. Now, when you look at these, you may not be sure what you're looking at, but again, just remember that you would find carbon atoms at these different corners. And then you know through dehydration synthesis in that previous picture that we'd have an oxygen in here and a carbon coming together. And then if you could look at the structure, you might have an idea that these are glucoses or something like that. It just depends on, but that's the idea. So again, remember, polysaccharides are then made up of multiple monosaccharides. Now here's a starch, and it actually has side chains coming to it. Now let's talk about lipids. Lipids are a lot like carbohydrates, except that they don't have as many hydrogens to oxygen. So it's not the hydrate anymore. And so they're actually difficult to dissolve in water. They still have carbons, hydrogens, and oxygens, but it's not the CH2O that we saw with the carbohydrates. So as you know, if you try to combine liquids with oils, the oils and the liquids don't dissolve together. There's a variety of different fats. Here are some of the more important ones that we find in organisms. Triglycerides, tri meaning three. So there's three fatty acid chains actually attached to a glycerol. And if you remember the glycerol, it's kind of like this glyceraldehyde. It is going to have a three carbon chains that come off that allow the fatty acid chains to attach to it. You'll see it in the next picture. Again, it's going to use dehydration synthesis to do so. But fats have all sorts of important functions. Obviously, they're important for energy. They also cushion organs. They provide insulation to help keep you warm. So there's lots of functions. The cell membranes, for instance, are made up of lipids or fats. Your hormones like testosterone, estrogen are made from a cholesterol fat. So here is our triglyceride before and after. So here is our glycerol. Again, it has those hydroxyl groups. So this would be a carbohydrate initially. And then as you'll see, the dehydration synthesis takes place, removing the hydroxyl group and the hydrogen and causing the carbon to join with the oxygen. And so we have triglyceride as the after effect. Again, the arrows going this way and that way means that the chemical reaction can go in either direction. Again, it's not shown in this picture, but there are proteins that we call enzymes, and they actually have an ACE at the end. That's a term that you'll see, like if you're talking about the enzyme that breaks down milk, we'd call it lactase. So anytime you hear the word ACE at the end, it usually means it's a protein that is an enzyme that speeds up chemical reactions. So hydrolysis would be the opposite. If you wanted to break this, your body wanted to break this down, hydrolysis would add water to do so. And so the byproduct, again, is going to be those three water molecules. Now, you probably have heard of unsaturated and saturated fats. Well, notice when you look at these three different chains, there's a little bit different chemical structure going on. In this case, the carbons over here are completely full, meaning that they have covalent bonds that are attached to different atoms. So this carbon atom is attached to this carbon atom through a covalent bond, this hydrogen atom, that hydrogen atom, and that carbon atom. So we call this saturated. But notice over here that this carbon atom has a double covalent bond with this carbon atom. And so there's not a hydrogen coming off of it. And so we actually call that, and there's no hydrogen coming off of this side. So we're actually calling that unsaturated. Saturated meaning full or saturated with hydrogen atoms. And then over here, we see that it is not. So unsaturated fatty acids actually seem to provide some health effects based on how being unsaturated can make the fats maybe have a different biological property and different temperatures and so forth. So this is, anyway, the difference when you talk about what a saturated fat is and an unsaturated fat is, this is what we're talking about, how there's less hydrogen bonds joined. But in doing so, there's a differences in some of the biochemistry and how the chemical reacts to the environment. Now here we have our phospholipid, and this time you see a glycerol backbone again, but notice there's nothing coming off of it, so it's not a triglyceride. There's only 2 fatty acid chains. Then we see a phosphate group that holds 5 bonds. So 1, 2, 3, 4, 5. So there's 5 bonds. One of the oxygens has a slightly negative charge because of that means that more of the electrons are spending time around the phosphorus atom, making this have, excuse me, I take that back. This oxygen is holding on to more of the electrons, giving it a slightly negative charge. My apologies. So the electrons are spending more time around the oxygen atom, giving it a slightly negative charge. And so that would be a kind of anionic, a negative charged atom. And then over here we have our nitrogen and some carbons. And again, this is shorthand. We know that this carbon would have hydrogens coming off of it. We're just keeping it simple by putting a subscript 3 here. And so what we found out in this chemical structure is that this side actually can dissolve in water easily, while these fatty acid tails, as you know, fats don't dissolve in water, don't. So we call this, when they don't dissolve in water, these fatty acid tails, we call it hydrophobic, meaning it's fearful of water. And then this side dissolves in water, so we call it hydrophilic or water loving. So water loving, water fearing. And this causes the chemical to face towards the water, this side, and this side to face towards the other lipids. And in doing so, it actually forms a cell membrane, as you'll see in our cell biology class, or maybe in the next slide or so too. But basically, this is what allows a cell membrane to form. Lots and lots of these chemicals. And again, you'll see when we talk about cells, it's a bit more like a soap bubble. And if you remember, soap is even made of, I don't remember exactly how it's done, but it requires fats and adding lye to it and so forth. Now, a lot of the lipids are also important for making cholesterol. Cholesterol isn't just bad, it's actually important for cell membranes, as well as making various hormones and vitamin D and estrogen and testosterone and so forth. What you see is these carbon rings. So when you see 4 interconnected carbon rings, you know it's a sterol. Again, shorthand would suggest that there's a carbon atom here that's showing a covalent bond going in these 2 different directions. So that means there's got to be at least 2 hydrogen atoms coming off of that carbon. While over here, because it's double covalent, this carbon atom has 2 bonds here, 1 bond here. That means, oh, it can only hold 1 bond in this direction. Anyway, different modifications of this 4 interconnected rings and side chains and so forth make it to be different types of hormones. And again, these are all done through biochemical pathways requiring enzymes to do so. Lipids have other functions as well. They can form the waxes that you find the waterproof feathers and leaves and fruits. So the waxy coating isn't just accidental, it's actually important to help prevent leaves from dehydrating and fruits from dehydrating, or keep the water from waterlogging a duct, so it can basically get the water rolling off its back, so to speak, and of course float, or help it to float. It's not the only reason it floats. Amino acids are our proteins. These are the structure and enzymes that take place in our body. This is the most biochemically important chemicals that your body will make or need to do all the different types of enzymatic reactions on structural structures like proteins and stuff like that. Obviously, you need the lipids to have the actual cell membranes. But when a fat or a lipid gets modified or a carbohydrate gets modified, it's done so via these proteins. They are made up of monomers we call amino acids, and they contain the atoms carbon, hydrogen, oxygen, nitrogen, and to a lesser extent, sulfur. And so this is the basic building block of proteins, this amino acid. And why do we call it amino acid? Well, it has an amino group, which is a nitrogen with 2 hydrogens coming off of it, and another one going to the carbon. So we know that nitrogen can hold 3 covalent bonds. And then over here we have a carboxyl group, and that has a carbon with 2 double bonds, an oxygen and a hydroxyl group. And again, of course, there'd be a covalent bond coming off the hydrogen group. Then you'll see the central carbon atom that's holding the nitrogen and the carbons together. And then you'll see a letter that says R. R means it just could be a variety of different types of side chains of different atoms joined together, carbon atoms. So alanine, for example, would have a carbon and 3 hydrogens coming off of it. That would be the R group. The cysteine would have a carbon, hydrogen, and a sulfur and a hydrogen. Tyrosine, look how complex that is. It has a carbon and plus this ring here of other carbon atoms and some hydrogen. So this is a really complex. And so you can see what happens when you join these monomers together. You get slightly different structure because of these different R groups, and they cause the enzymes to have certain functions. Again, how do we join these 2 amino acids together? We do so through dehydration synthesis. So we would remove the water molecule. Again, an enzyme is responsible for doing that. And when you eat your meat of a burger or something like that, or plant protein, you're going to drink in some water, and that water is going to do hydrolysis that breaks that amino, that covalent bond. We actually call it a peptide bond. So what happens when we make a covalent bond between a nitrogen and a carbon through dehydration synthesis? We call that a peptide bond. You can break down peptide bonds, again, as a covalent bond with hydrolysis. So again, the chemical reaction goes in both directions. Enzymes are involved in this process to speed up the chemical reaction, which is also made up of proteins and made up of all these amino acids. When you got a bunch of amino acids lined up, we call that a polypeptide because of the peptide bond. You have multiple ones, poly, we call that a polypeptide. Now, if you just have 2 amino acids joined together, we call that a dipeptide. And then we call them a free amino acid if it's just by itself. So again, there's probably around 20 something, I don't know the exact number of amino acids that our body uses. Some of them are essential, meaning that you have to get them out of your diet. Your body doesn't make them. I think that's around 5 to 7 or so. I'd have to look that up to be for certain. You would have to get it from your diet. And this can be depending on different animals. Some animals have their other types of essential amino acids or organisms. And then there's a lot of other amino acids that your body can actually make. So there's some that you have to get out of diet. And usually it's from protein sources that are very complete in amino acids, like eggs and meat, and some not necessarily vegetables, but certain combinations of beans and rice can help you to have a relatively complete amino acid profile to get you those essential amino acids. If you don't have the essential amino acids, you can't build the proteins that you need for your body. Now, if you have just a straight chain of amino acids, we call that the primary structure, a polypeptide. And then like a string, just imagine a string being a chain of amino acids. Imagine if I started coiling that string. like a shoelace or something. I started calling it. We scientists call that a secondary structure. There are hydrogens that kind of can hook up with oxygens and different things that have slightly different charges and form what you call hydrogen bonds. You may take a look and figure out what a hydrogen bond is by Googling it quickly if you're a little confused. And then we have tertiary structures. When you get your string, that's your all twisted up, that's your secondary structure or shoelace. And then imagine tying the rope into a knot of some sort, biochemical knot. That would be our tertiary structure. Now let's imagine you have multiple strings come together and you glue them all together. That would be our quaternary structure. Again, proteins have either a structural function, help your muscles to move, or a enzymatic function, causing chemical reactions to take place like dehydration synthesis and hydrolysis. There's also all sorts of other types of quaternary proteins that are important, like antibodies. Antibodies are what your white blood cells make that can attach to foreign particles in your body that we call antigens. So when you take the vaccine, your body builds up these antibodies that will then be ready to attack and identify a virus, not necessarily kill the virus, but identify the virus so your body knows to kill it and destroy it and not utilize it. Hemoglobin is a blood protein that has an iron attached to it, has a little heme group is what we call it. And so each one of your hemoglobin red blood cells would have around 1 million of these hemoglobins with 4 different quaternary structures that allow it to hold 1 billion or so oxygen atoms. There's other hormones like insulin that's made to help in regulating sugar and glucagon for fat metabolism. You have keratin that helps in hairs and fingernails. Fibrin and thrombin are found in your blood to help with blood clotting. Spiders, their silk is actually made up of protein and one of the strongest structures that we know of. And then again, all sorts of enzymes. Notice how you have the word ACE at the end. So maltase would break down sugars. Lipase would break down lipids. And then occasionally you'll see ones like pepsin, which is actually an enzyme that breaks down proteins. So enzymes are important for speeding up chemical reactions. Here we see enzymes joining these 2 substrates together. The substrates would be those 2 monomers, for one example that we've had in the past, or 2 amino acids. And the enzymes would bring them together and help speed up a chemical reaction or break down that chemical reaction. Because remember, these can go in different directions. It may be the same amino acid or maybe a different amino acid, but they help in speeding up a chemical reaction. We call this the active site that allows it to join together, and it often functions kind of like a lock and key mechanism. So because this structure is present, it brings these atoms together and helps in that sense speed up a chemical reaction versus these 2 chemicals bumping into each other accidentally and maybe undergoing a chemical reaction. So in other words, enzymes greatly speed up a chemical reaction. Then we have our nucleic acids. This is our DNA or RNA or adenosine triphosphate. These are important for making DNA, the blueprints of our cells found in our nucleus, or important for making up ribosomes or part of the ribosome. Ribosomal RNA is what we would call that, or transfer RNAs. And it's also important for making the fuel source called adenosine triphosphate, where you would have a sugar and a thymine and some phosphates coming off of it. And that is the fuel source for all of life, adenosine triphosphate. So it'd be the phosphates plus this thymine. Now, again, what are the nucleic acids made up of? Carbons, hydrogens, oxygens, nitrogens, and phosphates. So nucleotides are monomers. And then, of course, you join these together, you may have your DNA molecule, for instance. So here we have our DNA molecule, deoxyribonucleic acid. If you look at this, the sugar deoxyribose is critical for joining the nucleic acid with the phosphate. The phosphate and the deoxyribonuclease, the the deoxyribose, makes up the backbone of DNA. And then these nucleic acids are in the center. So I just want to make sure, and of course, I'm hoping you realize, of course, now at this point that you realize carbon atoms are present there. And so this is, again, shorthand. You know there's carbon atoms here. This is our nucleic acid base. We have 4 of them, by the way, if I failed to mention that. There's guanine, cytosine, thymine, and adenine. So these are the 4 nucleotides that are found in DNA, deoxyribonucleic acid. This is the nucleic acid part. Now, there's also a thing called RNA, ribonucleic acid, which is single stranded. We'll get into that later or in a moment. But in those cases, there's no thymine for the RNA. DNA has, again, guanine, cysteine, thymine, and adenine. RNA has guanine, cysteine, adenine, and uracil. So you'll see a uracil instead of a thymine. And you can see they're kind of similar, chemically speaking. So this is a little bit of what the DNA looks like. Here you have your nucleic acids. You see these are little dots. Those little dots represent hydrogen bonds. Basically, it's kind of like a weak covalent bond between a hydrogen atom. And then, and I can show you more of that later. But what you'll see is that cysteine always attaches to guanine and adenine always attaches to thymine. Always in quotes. I mean, obviously there can be mistakes made, but generally speaking, that's what happens. And then you'll notice the backbone, that's what we call this, the backbone of nucleic acids is made up of phosphates and sugars, deoxyribose sugar to be exact. And so notice that they're going in opposite directions. Here we have the phosphates and the sugar pointing this direction, and then notice that the sugars are pointing in the opposite direction. Going in the opposite direction. The letters are even upside down to represent how they're going in opposite direction. And then this all gets coiled up in a helix shape. And of course, it's stored in the nucleus. Now, RNA, ribonucleic acid, is single stranded, so you don't see the double strand. So if you look at this one, this is what we call a double strand. Here's a strand, here's a strand, so it's double strand. Here we call it single strand. And again, notice there's a uracil instead of a thymine. So this is ribonucleic acid. Okay, so here is some of the organic molecules that consist of a carbon-based core and some of their special groups. So this is how macromolecules are formed. So again, here's our amino acids. They're going to be made up of the amino group, and that's important for proteins, and the carboxyl group. So remember, that's what we saw on the amino acids. We see a carbonyl, which is our carbon with 2 oxygens coming off it. So anytime you hear the word carbonyl, you know it's a carbon with double bonded to an oxygen. Oh, here's our hydroxyl group. Remember how I was telling you about that? Here's our oxygen and our hydrogen. So again, these are what you see consistently in these different macromolecules. Hydroxyl groups you see in carbohydrates. So if you ever time you see in this OH chemical structure, you'll know that it's a hydroxyl group. If you see carbonyls, you know you're probably dealing with a lipid. If you see a carboxyl group, you know you're dealing with a protein. If you see an amino group, you know you're dealing with proteins. And then if you see this phosphate, and phosphate atom with 3 or 4 other oxygens around it, you know you're dealing with DNA or anisine diphosphate, anisine triphosphate, or anisine monophosphate. And then I just want to make sure that I didn't make a mistake on this. When I said anisine triphosphate, I was at that time was talking about the T molecule into phosphates. Anisine triphosphate would be 3 phosphates, a sugar, and an adenine nucleotide. So make sure you understand that. I may have said it wrong by saying thymine last time. Again, anisine triphosphate is the fuel source of cells, plants, animals, whatever, is the fuel source of your muscles working. Again, I may have said it incorrectly by saying thymine. This realizes anisine triphosphate. So it'd be this plus 3 phosphate atoms in this nucleotide. So that's anisine triphosphate. ATP, that's what the A stands for. Now, again, the organisms are primarily made up of 4 kinds of molecules. You are an organism. So you're going to be made up of proteins, nucleic acids, carbohydrates, and lipids. When you eat an organism, this is what you are digesting: proteins, nucleic acids, carbohydrates, and lipids. Now, they are termed macromolecules, and they constitute the structure, the building materials, the machinery, the enzymes to be large polymers. Again, we know what dehydration synthesis, so this part should be a review for you. But when we join together these amino acids, we remove the water. If we join together 2 sugars, monomers, we make water. And again, it requires an enzyme to do that. So you need enzymes, protein enzymes to do that. So this is, again, a little bit of review of what I mentioned earlier. And that will remove the water. So dehydration synthesis, we remove water. Hydrolysis, we add water. And again, it's going to use a protein enzyme because enzymes are made of proteins and speed up chemical reactions. So here is how the macromolecules get into these large structures. Here is our amino acids. We join a bunch of amino acids together. We have our polypeptides. And then ultimately we have protein structures like the cytoskeleton. Or we have a bunch of nucleotides, we join them together, and then we have our DNA strand that if that gets all coiled up, we have a chromosome. We have a bunch of monosaccharides joined together, we make our sugars, and then those are the starch grains that you can find in chloroplast. Remember, because the chloroplast is important for making carbohydrates by using sunlight and carbon dioxide and water to make starches and so forth. And then here's our lipids. Again, this is a fat cell holding a droplet of lipids. These are triglycerides. So this is how we store our fats in our body, is through triglycerides. And then again, I told you there's around 20 something amino acids. These are just different examples of it. Again, remember the R groups represent this side chain because we're going to have our amino group, our carboxyl group, a carbon, and then some side chain that we normally label R. Some will have different charges and some won't. All that has a big effect on what type of proteins we'll end up making. And again, remember I was telling you essential amino acids you have to get from your diet. And so here is dehydration synthesis. We removed a water molecule and we joined 2 amino acids together, forming a polypeptide chain or a linear chain. And then that gets rolled up, and then we have our secondary and tertiary and our quaternary, as I described in the previous slides. And that gives it all sorts of different functions, like enzymes are involved in breaking down and speeding up chemical reactions. So if you see the word protease, we know it breaks down proteins. If we see polymerases, we know that it produces nucleic acids, and kinases phosphorylate and add a phosphate to sugar. Some are going to be involved in antibodies and helping fight against snake proteins and toxins. And then some are involved in transporting, like hemoglobin is responsible for transporting oxygen in your body. So just take a look at this chart, read about it a little bit, and just realize how important proteins are. And then we have some that are involved in support, like your collagen is a type of protein that forms cartilage. Keratin is involved in hair and nails. Fibrin, as I told you before, is important for making blood clots. And then we have all sorts of regulating hormones that make, or regulation proteins that make up the hormones, as well as the fats. Remember how those are involved. There's milk proteins. There's all sorts of different enzymes and structural and stuff. Just take a little time to appreciate that. Realize your muscle fibers are made up of proteins. So take a moment, look at this slide to understand a little bit better. Again, I've already talked about the general structures of proteins, the primary structure, secondary, and tertiary. Primary is just that straight chain structure. This is again a little bit of review of what we talked about. Secondary is kind of coiled up. Tertiary, they're joining it in all sorts of shapes. And quaternary is usually adding other components together to make a more complex protein. So this picture here is representing these different levels of structure. The first level is this amino acid chain, polypeptide chain. The secondary structure is kind of coiled up in either an alpha helix or a beta pleated sheet. Tertiary structure, they're all kind of sticking and kind of forming a knot. There's going to be hydrogen bonds helping hold these together. And then quaternary structure, you have multiple tertiary structures coming together to make this enzyme that might function or something of that nature. And so that's kind of where this is all taking place. Now you can denature this protein. If you cook an egg, you'll take a nice structure of the protein and denature it so it's no longer in its proper conformation, meaning its proper shape. So when you cook an egg, you denature it, you put an acid, you denature it. And then those enzymes no longer function. Here's different types of proteins again. There's our structural ones. Here's our keratin, our silk from that you see in spiders, keratin for our hair and nails, fibrin for making blood clots. And then you see globular proteins that are important for like oxygen transportation and enzyme reactions, or for carrying other proteins around. So there's some proteins that are chaperones and they'll move substances around the body and make sure that they're all properly folded and connected. So they are very important for getting enzymes that have the proper shape. And then again, nucleic acids are important for storing genetic material like DNA and even RNA, which is kind of like DNA is the blueprints and RNA is like the photocopy. Again, they're made up of 5 carbon sugars with a nitrogenous base, that's the nucleic acids, and phosphates that make up the backbone. So here are our nucleotides, as I mentioned in previous slide. You have your phosphate group, you have your sugar, your deoxyribonucleic ribose sugar, and then the nucleic acid, which is can be either adenine, guanine, cysteine, or thymine when we're talking about DNA, and uracil if we're talking about RNA. So again, this is a little bit of a review of what I just described to you. DNA has deoxyribose sugar. RNA has ribose sugar. That helps explain the differences in the name. And then the nucleic acid that's joined causes it to be deoxyribonucleic acid or ribonucleic acid. Notice that the bases are different. As I already mentioned before, ribonucleic acid has uracil, while DNA has thymine, but the rest of it is the same, the adenine, the guanine, the cysteine. And then we have it single-stranded versus double-stranded. And then hydrogen bonds will hold either guanine and cysteine together or adenine and thymine together, but we call that double-stranded because of the 2 backbones. While the ribonucleic acid is a single-stranded structure. And it's kind of like the blueprints of that. And we'll get into more of that in future classes. Here's how complex it looks like once you remove some of the spaces. So this is what the atoms would look like. So these are all outer shells of atoms. So the electrons are on here. This is representing the outer shell. They're not these colors. That's just what the artists help you to see so you can visualize different atoms that are coming off of it. But this would be the sugar, or excuse me, this is the phosphate and sugar backbone. These are the nucleic acids that are hydrogen bound. So that's what's being shown in these pictures here. Again, carbohydrates are either going to be simple carbohydrates or complex carbohydrates. We've talked about the ratio before, so that helps you in understanding that. Monosaccharides are simple sugars, while disaccharides are going to be 2 monosaccharides joined together through dehydration synthesis. Again, this is a bit of review of what I described already. And these polysaccharides can then become things like starches, which are the energy sources for plants, glycogen, which is the energy source for animals, cellulose, which makes up the cell walls of plants and the structure that you can't digest, but certain things like termites and elephants, any animal that eats grass, they can have a symbiotic relationship and extra spaces that we call stomachs in quotes. or chitin that makes up the cell walls of fungi. And so this is where we find those things. Lactose is a disaccharide that we find in milk and specifically in cow. Starches, you can see, are polysaccharides found in potatoes and so forth. Cellulose makes up the leaves, and chitin makes up the structure of insects, plants, or excuse me, insects and fungi and so forth. And then lipids are going to be phospholipids and steroids. Here's our triglycerides. I mentioned that before. Here's how we store our fats in the glycerol backbone. And then some of them can be saturated and unsaturated. If they're saturated, of course, they have all their hydrogens filled, and if they're unsaturated, they have a double covalent bond, as I mentioned earlier. And that changes whether it's oily or not. And then phospholipids make up the cell membrane. And so that's what I mentioned again previously. And then here's our steroid, our 4 chains with the different side chains that make up the hormone itself. So I hope you have a better understanding of biochemistry from this lecture to help you move forward in understanding some basic cell biology. I appreciate your attention. If you're confused, be sure to watch some of it again, take notes, pay attention, and or watch other videos and YouTube videos to help you to understand it better. Anyway, I hope you have a good day. Take care. Bye.